My name is Ingvild Haugan and I'm a medical microbiologist. I work at St. Olav's Hospital and the Norwegian University of Science and Technology in Trondheim, Norway. I'm a member of a sepsis research group in mid-Norway and I'm doing a PhD where I retrospectively look at Staphylococcus aureus bacteremias. And the main goal is to try and identify any bacterial characteristics that are associated with phenotype or clinical phenotype. To do that, we performed whole genome sequencing of all bacterial isolates using Illumina technology. This poster is a part of a side project to that PhD, and the background for that side project is that a master student found an association between an adherence gene in the ESCMID family and a phenotype. But she correctly suspected that there was some misassembly of this gene due to several repeated sequences that these genes typically contain. So before proceeding with further genetic analysis, we wanted to try and investigate the reliability of the sequencing results. So to do that, we performed whole genome sequencing using Oxford Nanopore technology. And we also polished the Oxford Nanopore assemblies using Illumina data. So the results from that comparison is what's presented in this poster. First of all, the quality measures and typing results were quite as expected. The short read had higher per base quality and had higher coverage, and the long read had much longer contigs and higher N50. And after polishing, we saw that the long reads had about, on average, 20 base errors and a bit more than 20 indel errors per assembly. The SPA gene is also a gene that contains several short repeated sequences, and a correct SPA type or SPA sequence was found in about 86% with the short read technology and 100% with the long read technology, whilst sequence typing was similar between the two technologies. In regard to genes, we found that the polishing of the ONT assemblies did not result in any changes in the gene count. And we saw that the long read technology detected a higher number of genes compared to the short read technology. However, for the majority of genes, the results were quite similar. So it was a selection of genes that had a relatively high discrepancy between the two technologies. And an example of that, or a few of those genes are presented in the table in this poster. And one example is the delta hemolysin gene, HLD, which was detected in a higher number with short read sequencing compared to long read. However, the detection of the presence of the gene in an isolate was only in about 85% with short read technology compared to almost all isolates with long read technology. We haven't yet looked into the reasons behind the discrepancies for certain genes, but I believe that there's not one explanation or it's not the same explanation for all types of genes. So some genes may have several repeated short sequences that we know is difficult for short read assembly. Other genes may have high genetic identity with similar genes or other genes in a group, such as the enterotoxin genes. And there are also genes that are very short that may actually cause problems with long read assembly, such as the ERM-C gene. So I guess the take-home message is that it's important to really understand and know the strengths and weaknesses of the sequencing technology one wants to use for one's research project to make sure that it can answer the research question one has. And also to know that the differences between short and long read goes beyond base quality and contig length. And perhaps if one wants to do a more comprehensive genetic analysis, a combination of the two technologies may be a better solution. So for this poster, I want to thank the master student Nora Berg Blomqvist, who did the analysis that led us to this side project. I want to thank my supervisors that are co-authors of this poster and also the other co-authors. and both St. Olav's Hospital and the Norwegian University of Science and Technology that has helped me with funding for the project.